This is a very important topic for digestive systems for the MCAT. If you like this type of content, check out the link in my bio to my self-paced course. It has over 500 pages of typed out explanations for high-yield Chem Phys and Biochem content, as well as a downloadable schedule that walks you through how to prep for your entire MCAT process. We're going to look at how does glucose and amino acids get absorbed in the small intestine. We're zoomed in on the small intestine and notice that right here is the lumen. That means that on the other side of this, we would have more cells that make up the wall of the other side of the small intestine. So in this area right here, that is where the food is actually traveling through the intestine. The way that sugar and amino acids are absorbed is through a similar mechanism called secondary active transport. Now let's look at what that is. Secondary active transport starts with a regular sodium-potassium ATPase pump. So right here, ATP is used and it's burned up, and that is used to pump 3 sodiums against their concentration gradient from the inside of the cell to the outside of the cell. And then 2 potassiums are pumped from the outside to the inside of the cell. Then that sodium that's pumped out is going to move back into the cell through a different channel. As it moves through that channel, it's going to be moving with its concentration gradient because there's more sodium in the lumen of the small intestine. Since it's moving down its concentration gradient, that is going to be releasing energy. That release of energy is going to be used to take this glucose and move it across the membrane of that small intestine cell. There is a higher concentration of glucose inside of the cell than outside of the cell. So the extra energy that's released by sodium moving into the cell is actually necessary to move glucose into the cell. Without that extra energy, glucose would have no way of moving against its concentration gradient. We call that secondary active transport because the ATP that was actually used only happened in that first step, and we use the sodium that ATP pumped out as the source of the energy to move the glucose. For amino acids, the process is the exact same. We just use a different channel, but sodium does the same thing where it moves into the cell with its concentration gradient that releases some energy. That extra energy is used to move amino acids against their concentration gradient into the small intestine cell. Now let's look at the next cell over here. Once the glucose and amino acids are inside of that cell, they're going to be moving from the cell into the capillaries. This movement is with their concentration gradient because there's a high concentration of both of these inside the cell, so it doesn't take any energy for them to move from the cell into the capillaries. And then the capillaries are going to take those molecules to the liver.